Hello. In this video, you'll see how to make an IGV figure for your Unit 1 bioinformatics project. We'll look at how to download the files needed to view a genome and real sequencing data in IGV, and then we will look at how to turn that data into a figure for a single gene where individual variants are viewable by zooming in to the region of interest. The first thing that you'll want to do is open up two tabs, one with Galaxy with your history showing on the right-hand side, and the second with NCBI so that you can see the Lactobacillus reuteri genome and you can get the data files from the genome. So let's start with the genome data files. We're going to download the FASTA file, so download sequences in FASTA format for genome, and the genome annotation file, GFF. Both of these files come from NCBI in a gun-zipped GZ format, so we can use an extractor like extract.me and bring these files into the website. And save a copy of the output. So we don't want to save all as zip. We want to click on the output that gets created, S38 SU, download the unzipped output, then get the second file, download the output. Now I'm going to show both of these in Finder so that or in the File Explorer so that I'm able to rename them. So the S38 one was my first one. This is going to be our lacto reuteri.gff. And our second file, the S29 file, is going to be lacto reuteri.fasca. So now we have our two data files that we need to have a viewable genome in IGV. We can click on IGV and open it up and create a .genome file. In order to keep track of all the files, rather than leaving the FASTA and the GFF file in my downloads, I'm actually going to create a new file on my desktop just so that I have access to these. I'm going to copy these files, go to desktop, and create a new folder. The new folder will be IGV Figure for Unit 1. And I'm going to put my files that I need to make the genome. In IGV, there are two different parts of the menu that we use to load data and we use to load genomes. To load genomes, we want to go to the Genomes tab and we want to create a .genome file. I'm going to call the unique identifier biogastrus. That's the name of the pills. And the descriptive name will be the name of the organism. Select the FASTA file. And select the gene file. And say OK. It will create a new biogastrus.genome. I'm going to make sure that that goes into the same folder, IGV Figure for info for Unit 1. And now I have on the pull-down menu the one chromosome that is in that FASTA file. And it should see that I'm in the right genome because I should see a chromosome that is approximately 2 megabases, 2 million base pairs in length. Now I'm ready to load data. So I'm going to go back to my browser, go to my Galaxy tab, and the data that I want to be able to view in IGV is the sliced BAM file. For the example history, that's step 9. For your history, it should be a later step, maybe step 12 or step 14. I'm going to click on the slice and the download button, and I need to get both of these files. The dataset file will be a .bam file. the BAM index will be a .bai file. Now that I have both of these files, I can show them my folder. I can drag them to the new folder that I made for making the figure. Now in IGV, I'm able to load not just the genome, but the file. And I don't have to do anything with the file names. If I download them from Galaxy, they should be given names that will work for IGV. If I give them different names, I just have to make sure that both the BAM and the BAI file have the same name, because I'm only going to click on the BAM file, and then IGV is going to look in this folder for the same name file with a different extension, .bai. So I open the BAM file, and when I zoom in to most places in this genome, I will see no data. And that's because this is not the entire genome's alignment. This is just the one gene in the genome that I sliced the data to. If I type the location of the gene, and these gene locations, the start and stop base for your gene, are in the Unit 1 gene assignments Excel sheet, I'll be able to see the actual data. So this is the figure that I'm trying to make to show all of the aligned reads for my particular region of interest. You can see that there are several variants that are being flagged by colored stripes in the histogram of the sequencing data. And this is the figure that you'll make for your first Unit 1 project. Good luck, and let me know if you have any questions.